My name is David Kastner, and we're doing a project on working with beta hairpins and unnatural amino acids. And the purpose of this project is, in the last 10 years, one of the key advances in drug formation is the utilization of peptides. And the key with peptides is they're incredibly specific because they're naturally used in the body. And the benefit there is they are specific and non-toxic at the same time. However, the problem is, since the body is evolutionarily designed to handle peptide drugs, they're degraded very rapidly by a protein called proteases. So the purpose of our project is to design a new class of peptide that is resilient to proteases and resistant to being broken down. And so if we look at this image over here, this in general is the protease. And this right here is the peptide that I was talking about. And so normally in the body there are 20 different amino acids, and these are made by all organisms. However, we discovered another amino acid that was found in a deep sea sponge. And this type of amino acid is resistant to proteolysis. And if we insert it into a beta hairpin, the beta hairpin becomes more well formed. And so if we look at these two figures right here, this is the control peptide that normally forms the beta hairpin. You can see that it's not quite as rigid. There's waves in the beta hairpin formation, whereas if we put in a dehydroamino acid, it becomes much more rigid. So that is the advantage of these rare amino acids, that if we insert them into normal peptide drugs, they become more rigidly folded and they become resistant to degradation by proteases.